Good morning. Thank you so much for giving me the opportunity to listen to Globavir's story. My name is Shalabh Gupta. I'm founder, president, and CEO of the company, and it's my pleasure to walk you through our company's story, where we are scientifically as well as in business side for both our drug as well as diagnostic portfolio. Slide number 2, I'm going to go through some forward-looking statements. These are statements which you should review before making any investment decision. Slide number 3, it gives you a table of content. Slide number 4 is where I am, and I will walk you through what is so special about the company, what is so exciting about the company. So Globavir is a company that is focused on infectious disease. We have a late-stage drug that is starting phase 2 study. It was developed at Stanford. We have a global exclusive license from Stanford. And the drug addresses a orphan disease market in the United States, but has a potential to go for priority review voucher, which has fetched many hundreds of millions of dollars to other companies. We also have a diagnostic portfolio on which one of our diagnostic tests is already commercial. It's approved in Europe as well as in India, and we just received our first 10,000 test order, netting us around $80,000 revenue. We have other diagnostic tests in the portfolio, and with that, the company has both the drug as well as a diagnostic test that helps us identify patients who are the right patient for the clinical trial. Moving on to slide number 5, it gives you a little bit of history about where we are coming from. I started the company leaving Genentech in 2010. First set of assets were oncology, which we licensed from USC, University of Southern California, and LA, and sold that to Sorrento Therapeutics. In 2015, Sorrento became an investor. Subsequent to that, we have raised money from Stanford's 2 different funds. We were also selected as the only biotech company in 2014 to be part of Stanford's StartX, which is a Stanford's accelerator program. And we have also partnered our diagnostic tests with Bio-Rad, which is a global diagnostic company with several billion-dollar market cap. So our technology, our company's pedigree, has multiple different places which give you validation, independent validation, as well as some comfort to know that our technologies, people we have worked with, these are the top-notch people in their field. We have been successful. I'm looking at slide number 6. We've been successful both in licensing our assets, which I've already shared with you, USC, the first oncology set of programs, and Globavir's program. They're all in license from Stanford University. But we have also been successful in commercially developing our programs as well as partnering. Besides Sorrento, we have partnered a program with Bio-Rad. And as you think about the company's portfolio and moving forward, having a drug that affects several million, hundreds of millions of patients, it's important for us to keep in mind that Globavir as a company would have to seek other relationship, other partnership as we move forward to bring our program to provide these drugs and diagnostic tests to patients across the globe. Slide number 7, looking at the partnership related to our drugs. So early on, our technology was developed at Stanford University. We have a global exclusive license, but we have ongoing relationship with the Stanford in which we work with our colleagues at Stanford to develop the drug, also look at the potential ways that we can run successful clinical trial as well as run through the regulatory approval. We also partnered with Duke NUS, which is Duke National University of Singapore. It is one of the premier teaching hospitals as well as affiliated with their medical centers. Dengue affects people primarily in Southeast Asia and Latin America. So finding a partner in Southeast Asia was natural for us. We are fortunate to have the support from many different physicians and scientists at Duke NUS, and also a collaboration that will allow us to start a clinical trial in Singapore. Moving on to slide number 8, next set of inflection points are several. This year we plan on starting phase 1b/2 clinical trial for dengue fever. We also have ongoing programs which are evaluating the same drug, GBV006, for other indication, primarily the next indication after dengue is chikungunya. We are looking to get other diagnostic tests, which is Mulglobe, to get approval and start to sell that in commercial markets latter part of this year. Slide number 9 gives you an overview what the drug is. GBV006 is a combination of two drugs, 001 and 002. These two drugs work in synergy, which means these drugs don't just have additive effect, 1 plus 1 equals not 2, but more like 5 or 7 here. And the drugs work by working on the host cells, which is human cells. Our drugs, there are two drugs, they work in combination. One of them helps to block the virus entry into the cell, and the other one blocks the virus multiplication once the virus is inside the cell. Therefore, once given in combination, the drugs can block the new viruses, new infection to go inside the cell. And since it's a treatment that we are pursuing first, there are some viruses that are already in the cell, and instead of giving them a chance to continue to create damage, the second drug blocks the virus multiplication once the drug is inside the cell. Again, this is a drug that was developed at Stanford University. We have a good set of data in animal models, which I'll show you in the next few slides. And we have a global exclusive license with Stanford. Because the drug works in host cells, going to slide number 10, what it does is that gives us an opportunity to have a broader spectrum coverage. When I say broader spectrum, what it means that the host mechanism, which is the virus entry into the cell and multiplication within the cell, is the same host mechanism that is common to other viruses, not just dengue. It is also a mechanism that is found in Ebola virus, as well as in other viruses like chikungunya. And once we develop a drug in treatment of one disease, we can pursue the data and run additional trials for other indication. And this is what I mean by having a broader spectrum approach. But because we are working on the host cells, not working on virus cells, we also have an opportunity to have less resistance. As you know, diseases like flu, having a vaccine in the beginning helps, but then virus mutates and the vaccine becomes ineffective. But once you have a drug that is not working on, it's not dependent on the virus, but is working on human cells, by making it harder for virus to get entry or harder for them to multiply once they're inside the cell, that means we have less susceptibility to any of these resistant mechanisms. The cartoon on the right gives you a pictorial view about how the virus gets inside the cell, what's the mechanism, and endocytosis is a scientific term that is coined for these type of viruses to get inside the cells. Slide number 11 gives you a nice overview of what happens when the drug is given to the mice. AG129 mice is a gold standard for the dengue infection, and we have tested our drug in not just AG129 mice, but other mice models as well. We show you the data from here. What it shows you that once the mice are given lethal dose of dengue virus, which means there are two sets of mice, one are given the dengue virus and they are not given the drug. The one are given dengue virus infection and they are given the drug. Those that got the dengue virus infection but were given no drug, they all die 100% mortality after first 4 days. Those that were given the drug, they were given the infection virus load and were given the drug, they had 100% survival. And these viruses not only survive, these mice not only survived, but also had no loss of appetite and continued to have fully functional activities continuing for several days after the infection, for several days of monitoring. With that, I'm going to move on to slide number 12. What we are showing here is the data that these drugs, GBV001 and 002, have been tested both intraperitoneally, IP stands for intraperitoneal, as well as orally, and our effects these drugs have is consistent both regardless of the route of administration. The drugs, GBV001, 002, were given both before the virus lethal dose was given to these mice, as well as given after mice were given the inoculum of lethal dengue virus, and to see, does it make a difference whether we give the drug only after the infection has happened, or does it matter if we give the drug even beforehand? And what we are showing here is that regardless of when you give, which one would be prophylaxis. So in that case, you're giving somebody, if you translate this data into human data, it will be giving a drug to somebody before they get the infection. and the other one is you give them the drug after they get infection. So the first will be prophylaxis, the other one is treatment. In the animal model, we are seeing the drug works both as a prophylaxis as well as a treatment. For a variety of reasons, which would be hard to explain in this small presentation, a short presentation, we are currently pursuing the drug as a treatment, not as a prophylaxis, but one of the application is beyond going through the initial treatment approval is to pursue prophylactic treatment. The drug going into phase 2 clinical trial, it will cost around $2 million or so. We are running this trial in Singapore, as I mentioned earlier. We have announced a partnership. We plan on running this in a partnership with Duke and US. The phase 3 trial, based on what we have had initial discussion with regulatory agencies, will cost us around $6 million, and this is based on around 200 patient data. Since there is no available treatment, we believe the regulatory pathway to get a drug approved is to our advantage, because many regulatory agencies throughout the world are very concerned and they are interested to find a treatment. If there was a treatment approved, we will have to compare ourselves against the standard of care. The standard of care currently is no treatment except giving symptomatic treatment, which I already mentioned to you, is IV fluid, Tylenol, and in some cases platelet transfusion. This talks about the timeline, which I wouldn't go more in detail. I've already talked about it, that we are starting a clinical trial towards the end of this year. We protect our intellectual property, which is one of the key component or thesis of investment in this company. We have already a granted patent that was granted in 2016. We have filed one of the patent, which is a fixed dose combination patent that was filed late last year. And then we are also working on different types of formulation, including injectable formulation of the drug that we plan on filing in the near future. A little bit on the diagnostic side here, we are on slide number 18. We are looking at our diagnostic portfolio. There are 3 different tests, which is also on our website: PanGlobe, DenGlobe, and MulGlobe. Instead of going into names, let me give you a big picture. What's the idea here? Pan makes it somewhat easier to understand that it is taking all different serotypes, which I shared with you earlier. Dengue has serotype 1, serotype 2, serotype 3, and serotype 4. This test helps one identify a patient has dengue. So it's essentially yes or no test. This test is approved in Europe. We have a CE mark. It was also approved in India. And we've just received the order for our first test, first 10,000 test, which I shared with you in the beginning of the presentation. DenGlobe is a test that is basically identifies which serotype patient has. So it can help the identification of serotype as well as quantification of viral load. And the MulGlobe is a multiplex test that helps identify many different pathogens with a single test. We are currently looking at 4 different pathogens, and these pathogens are dengue, leptospira, malaria, and chick. We did a partnership, which I shared with you earlier, with Bio-Rad, and Bio-Rad sells their test PanGlobe as an assay, but we still sell PanGlobe as a standalone test on our own. Our team consists of a small team with Chief Medical Officer, Dr. Claude Gordon, who has been part of dengue program before, and the Scientific Advisory Board has people who have done this work before. Strategic Advisory Board, again, consists of people who bring the business background, as well as our IP partner from Wilson Sonsini on our Strategic Advisory Board. And that is a way we can protect our company, both from intellectual point of view, intellectual property point of view, as well as from thinking about business strategy. On a board, we have Dr. John Ryan, Dr. Sandeep Lamas. John has been Chief Medical Officer of Cadmon. Sandeep Lamas has been part of a few different NASDAQ-listed biotech companies as their member of board of director. I'm looking at slide number 23, and this gives you a background about the companies that have been acquired in infectious diseases before. And you can see infectious disease is a niche indication. There have been very good successful exits. Slide number 24 also talks about the types of companies that are currently listed in capital markets. And that provides an opportunity for us to be able to take the drug into clinical trial, be able to create a treatment for patients that have the patients who have no treatment available. At the same time, be also able to create a good return for our investments, for our investors. With that, I come to the end of the presentation. I look forward to hearing your questions in live Q&A, and thank you again for giving me the opportunity to present Globavir to you. Thank you so much.
Okay, we're now opening the line for our Q&A session with Globavir. And with us today, we have the CEO, Shalabh Gupta. Welcome, Shalabh.
Thank you. Thank you for having me.
Our first question, can you talk about the status of your phase 2A studies for dengue fever, and have you started recruiting patients for this clinical trial? And do you have locations picked out for where you're going to be conducting the trial?
Thank you. For phase 2 clinical trial, we are conducting a clinical trial in Singapore, and the location is going to be affiliated hospitals, but primarily it will be Singapore General Hospital, which is an affiliate of Duke National University of Singapore, Duke and US. That is where the clinical trial is going to start. We had a meeting which is like a pre-IND meeting before starting the clinical trial in Singapore with the Singapore's HSA. HSA is equivalent to the US FDA, and they've agreed with us our proposed clinical trial protocol. So we plan on starting the clinical trial in the next few months. I would be able to give you more specific outline within a matter of a few weeks because we are having the final conversation with them to start the trial.
Very good. Thank you, Shalabh. And our next question, can you provide some additional color on your non-exclusive global license with Bio-Rad for the Pan-Globe assay?
So Bio-Rad has started selling these Pan-Globe assays as a part of a multiplex test globally, and Bio-Rad launched the test later part of last year, 2017. And at the moment, the test is widely available in various parts of the world, including the U.S. Our Bio-Rad is currently pursuing this test as a research use only called RUO, and we started receiving royalties from Bio-Rad.
Very good. Thank you. And our next question, can you talk a little bit about how your assay, or how you differentiate yourself from other biotechs in the space in regards to the detection of dengue fever?
A couple of points in this question. So there are multiple tests out there for dengue diagnostic. Most of them, not all, but most of them are focused on NS1 antigen detection test. And NS1 antigen detection test, regardless of who is doing, which company is pursuing it, their sensitivity and specificity is quite lower. The other types of tests are antibody tests, and they are slightly better than antigen detection tests. What we have, a Pan-Globe assay and Pan-Globe test, these are RT-PCR, so these are RT-PCR-based tests. By default, instead of relying on an antigen or antibody, which are prone to cross-reactivity, also false positive, false negative, RT-PCR tests are better in terms of sensitivity, better in terms of specificity. But our specific test within the RT-PCR test is the test available by other diagnostic companies. Head-to-head, the test is better in terms of sensitivity and specificity. Additionally, currently there is almost no test that we are aware of that can detect dengue virus after first 5 days of fever. We have Pan-Globe that can detect this virus up to first 10 days of fever. So from both sensitivity specificity point of view, compared to other RT-PCR tests that are available, our test surpasses by far. Additionally, we also have a test that can detect dengue virus after first 5 days, and the clinical relevance of that detection is that we can better predict patients who are going to have complications by being able to detect those patients who would otherwise not be diagnosed with dengue.
Very good. Thank you. And our next question, can you talk a little bit about what major milestones investors might be able to expect over the next 6 to 12 months?
I would call out your attention to 2 or 3 milestones. The first major milestone would be starting the phase 2 clinical trial, as we talked about this earlier. The second milestone would be getting the approval or validation of our second diagnostic test. One of them is multiplex test called Globe. We are currently working with various agencies as well as running clinical trials to be able to validate that. So I anticipate both these milestones to be able to meet within the next 6 to 9 months. And last but not the least, diagnostic is not our core focus, but we have the third test called dengue that is also in the process of going through the validation process and registration approval.
Very good, thank you. So quite a few things for investors to look forward to for this year. And that actually was our last question that had come in, but we have a few more minutes remaining. I guess just in summary, there was a lot of great information covered in the presentation as well as here in the Q&A. But what's the key takeaway for investors? I mean, why should they have interest in the company today?
Globavir's lead drug is GBV006, which is potentially the first treatment for dengue, where dengue affects many millions of people outside the U.S., but from a U.S. point of view and investors' point of view, this is an orphan disease that falls in the U.S. FDA criteria. And also, potentially, GBV006 can be a drug for eligible for priority review voucher. If we are able to get PRV, there is a huge upside financially for the company. Regardless of PRV, as an orphan disease, dengue treatment is something that many bigger companies are focusing on in terms of prevention with vaccine, and we have a treatment that can be potentially the first treatment out there in the market. So there is both opportunity to be innovative as well as doing very well commercially for our investors.
Very good. Very exciting story. Again, this was Globavir. We were speaking with CEO Shalabh Gupta. You can find out more information here at RedChip.com about the company.